Immunology Insights, Basics and Beyond, Chapter 40, Hematopoietic Stem Cell Enrichment in Graft Engineering. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our last episode, we explored the fascinating idea of immune tolerance, training the immune system to accept transplanted tissue without the burden of lifelong immunosuppression. But how can we support or even accelerate that vision on a cellular level? Today, we take a closer look at a key tool in the future of transplantation, hematopoietic stem cell or HSC enrichment and graft engineering. Now, you might think of a transplantation in terms of solid organs like kidneys or hearts, but bone marrow and stem cell transplants are transplants too. In fact, they represent one of the earliest forms of transplantation therapy. And more importantly, they serve not just to replace diseased blood cells, but to reshape the recipient's immune system from the ground up. That's where graft engineering comes in. By refining what's in the transplant, so removing harmful cells and enriching helpful ones, we can improve safety, enable tolerance, and even tailor therapy to individual patients. In this episode, we will explore how stem cells are characterized and selected, what clinical benefits they bring, and how they are being modified for the future. So, let's talk science. Why do hematopoietic stem cells matter? Well, hematopoietic stem cells or HSCs are rare, multipotent progenitor cells residing in the bone marrow and, to smaller extent, in peripheral blood. They are characterized by their self-renewal capacity and ability to generate all blood and immune cell lineages, including T cells, B cells, natural killer cells or NK cells, monocytes, granulocytes, and platelets. A key surface marker used to identify and isolate these cells is CD34, a glycoprotein expressed on early hematopoietic and progenitor cells but absent on mature blood cells. CD34 positive selection ensures that the transplant contains a concentrated pool of true stem cells with regenerative and immune rebuilding potential, while minimizing contaminating immune cells that might trigger complications like graft versus host disease or GVHD. Accordingly, there are quite some enrichment and selection techniques. To maximize safety and therapeutic potential, HSC grafts undergo careful enrichment and selection using advanced laboratory methods. First, we have CD34 positive enrichment. Techniques like magnetic activated cell sorting or MAX and fluorescent activated cell sorting or FACS exploit the presence of CD34 on the stem cell surface. Magnetic beads or fluorescent antibodies bind to CD34, allowing the cells to be separated from other blood components. The result is a high-purity HSC population that enhances engraftment and reduces the risk of immune-mediated complications. Second, T-cell depletion. Mature donor T cells can cause GVHD, especially in haploidentical, so partially matched transplants. Using depletion strategies, these T cells are selectively removed while retaining the HSCs. However, complete removal of T cells carries risks, such as a slower immune reconstitution, higher susceptibility to infections, and sometimes graft failure. Therefore, protocols are designed to maintain a balance between safety and immune competence. Third, co-infusion strategies. Some protocols involve combining CD34 positive HSCs with immunomodulatory cells such as regulatory T cells or Tregs or mesenchymal stromal cells or MSCs. These supporting cells help reduce inflammation, prevent alloreactivity, and improve the chances of stable graft acceptance, particularly in experimental solid organ transplant settings. Of course, there are many clinical applications. So why are HSC used in clinical settings? Well, HSC transplants offer something unique, the ability to rebuild the immune system from scratch or to correct underlying immune defects. So their clinical applications go far beyond than just cancer treatment. First, allogeneic bone marrow transplantation. In patients with leukemia or other blood cancers, HSC transplantation allows for complete immune system replacement, eradicating diseased cells and reconstituting healthy blood cell production. CD34 positive enrichment is particularly valuable here, enabling reduced intensity conditioning regimens that are safer for older patients or those with comorbidities, while still maintaining robust engraftment and reducing GVHD risk. Second, solid organ transplantation. Some cutting-edge protocols combine kidney transplants with HSC transplants from the same donor. The aim is to induce mixed chimerism, a state where donor and recipient immune cells coexist harmoniously, leading to a long-term immune tolerance and reducing or eliminating the need for lifelong immunosuppression. Third, gene therapy for inherited immune diseases. In genetic disorders like severe combined immunodeficiency or SCID, or Wiskott-Aldrich syndrome, a patient's own HSC can be harvested, genetically corrected, and reinfused. This approach offers a permanent cure by correcting the immune defect at its roots, without the risk of GVHD associated with the donor-derived transplants. For sure, this field has its challenges, but comes also with innovations. Despite their promise, HSC therapies come with technical and biological challenges. First, we have engraftment failure. Successful transplantation requires a sufficient number of high-quality stem cells to establish a new, functional blood system. Factors like immune rejection or insufficient conditioning of the recipient's bone marrow can lead to failed engraftment, particularly in mismatched or haploidentical donor scenarios. Second, we have GVHD versus graft versus leukemia, so GVL. While T-cell depletion reduces the GVHD risk, donor T-cells also help eliminate residual leukemia cells, so the GVL effect. Striking the right balance between reducing harmful alloreactivity and maintaining anti-leukemia immunity remains a central challenge. And third, ex vivo expansion. HSCs are notoriously difficult to expand outside the body while retaining their stemness. Current research focuses on molecules like SR1 or stem regenin 1, which inhibits the aryl hydrocarbon receptor to promote stem cell self-renewal, and notch ligands, which help maintain undifferentiated states during culture. As for future perspectives, the future of HSC-based graft engineering is advancing on multiple fronts. First, CRISPR-engineered HSCs. CRISPR-Cas9 gene editing is being used to create tolerogenic or immune-evasive HSCs, potentially leading to universal donor grafts that are compatible with any patient without the need for aggressive immunosuppression. Second, in situ programming. Emerging techniques aim to deliver transcription factors or reprogramming cues directly into the patient's bone marrow, effectively editing the immune system on site rather than relying on ex vivo manipulation. And third, graft engineering kits. Standardized off-the-shelf kits are being developed to streamline CD34 positive selection, T-cell depletion, and cell add-backs like Tregs. These tools bring precision medicine closer to the clinical frontline. To conclude, HSC transplants are not just treatments, they are immune system resets. By providing a fresh source of blood and immune cells, they can cure otherwise lethal diseases, reprogram immune responses, and even pave the way for tolerance in organ transplantation. With the rise of graft engineering, HSC-based approaches are evolving into customizable therapies tailored to the unique needs of each patient. As we push the frontiers of transplant science, these strategies may allow us to design the perfect graft, safe, effective, and long-lasting. In our next chapter, we take a moment to step back and honor the scientific legacy that made all this possible. We'll explore the life and discoveries of Sir Peter Medawar, the Nobel Prize-winning immunologist who laid the groundwork for transplantation biology and changed medicine forever. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So see you next time and thank you for listening.